Hi everyone, so today we're going to be looking at regulation of blood glucose and how our body maintains homeostasis in relation to the amount of glucose in our bloodstream. So firstly, what is glucose? It is our primary source of energy. It is the simplest form of carbohydrates, and we get glucose through eating carbohydrates. So for example, if you're eating a potato, you're going to be ingesting starch molecules. Your body is going to digest those starch molecules and produce glucose molecules. Why do we need glucose? It is an input for cellular respiration, and all our cells need glucose to produce energy. So this is a simple equation for cellular respiration, which you will go into much more detail of in year 12. I don't need you to know this equation. All I need you to know that is all our cells undergo cellular respiration, and this is the way that we produce energy. And one of the inputs for cellular respiration is glucose. So glucose is very, very important. All our cells need it. So let's look at blood glucose. So once our carbohydrates have been digested, they've been broken down by enzymes, they enter the bloodstream as glucose. And our glucose levels in our blood can be measured. So our normal blood glucose levels is about 4.0 to 7.8 millimoles per liter. So we have our target or our optimal range. Above this optimal range, we will have hyperglycemia. So hyper means high. Glyco, which we've looked at before, refers to carbs, right? And then emia is a suffix that refers to the blood. Excuse my writing here. So hyperglycemia means high carb or glucose. Glucose. I'm so sorry about my writing. High glucose in the blood. High glucose in the blood. So hypo is low. So hypoglycemia is low glucose in the blood, or low levels of glucose in the blood. And you have some images down here that explain that. So we want our normal levels to be this 4.0 to 7.8 millimoles. Below that range, we have hypoglycemia, not a lot of glucose in the blood. Above this range, we have hyperglycemia, a lot of glucose in the blood. So let's have a look at glycogen now. So if glucose is not used for cellular respiration, it can be stored in the liver or your skeletal muscle as glycogen. And so glycogen is just a long chain of glucose molecules. As you can see here, we've got one glucose molecule, and the glycogen is made up of multiple of these glucose molecules. This is important, right? Because we know that glucose is an input for cellular respiration. It's very important in producing energy, in keeping us alive. So if we have excess glucose, we want to store it. We don't want to get rid of it. So we will store it either in the liver or the skeletal muscle as glycogen. When we have high blood glucose levels, so hyperglycemia, this triggers something called glycogenesis. And that is the joining of those glucose molecules to form glycogen to then be stored. Right, so we have excess glucose or high blood glucose, we're going to have glycogenesis. Remember, glyco refers to carbohydrates, and genesis means to create. So we are creating glycogen from that excess glucose in our blood, and we're storing it. If we have low blood glucose levels, so hypoglycemia, this triggers glycogenolysis. So lysis means to essentially to break down. Right, so we're breaking down glycogen. So the word is right here, glycogen. We're breaking down glycogen into those glucose molecules to re-enter the bloodstream because we have low blood glucose levels. So we need to increase the amount of the glucose in our bloodstream. So we're going to go into this in a little bit more detail, but I hope you understand the importance of glucose and the importance of glycogen in terms of regulating blood glucose levels. So let's look at that now. We have our stimulus response model here, right? For homeostasis in terms of blood glucose regulation, the receptor and the modulator is something called the islets of Langerhans, which is a tissue in your pancreas. So the receptor and the modulator are the same thing. So let's have a deeper look. Stimulus, we've got a change in blood glucose levels. Your receptor is your islets of Langerhans tissues in the pancreas. Your modulator is the same. The effector is various cells and tissues. We're going to get to that in a moment, and then our response is to either increase or decrease the blood glucose levels back to normal and maintain that stable, optimal blood glucose range. So let's look at the pancreas. The pancreas is super important. This organ is the key organ in maintaining blood glucose levels. We've already looked at the pancreas in terms of the digestive system and the endocrine system and how it's involved in those two systems. And we're going to look at it again now and how it's involved. For digestive system, it produces and secretes enzymes used in digestion, and then in the endocrine system, it produces and secretes hormones for regulating blood glucose levels, and that's what's important today. So let's look at the role of the pancreas, right? The islets of Langerhans. This is a tissue inside the pancreas. The islets of Langerhans are made up of alpha and beta cells, and these alpha and beta cells release hormones. Those hormones are, so alpha cells produce glucagon and beta cells produce insulin. Right, it's important you know that the islets of Langerhans are a tissue in the pancreas, and the islets of Langerhans are made up of alpha cells and beta cells. Alpha cells produce the hormone glucagon, beta cells produce the hormone insulin. So this is just another breakdown of the pancreas, the islets of Langerhans, the alpha and beta cells, what they release and what they produce. So we know that the alpha cells secrete glucagon, this hormone. This increases blood glucose levels by breaking down glycogen. So glucagon leads to increasing glucose levels by breaking down glycogen. Beta cells secrete insulin, and they decrease blood glucose levels by converting that glucose to glycogen to be stored. It also promotes the uptake of glucose by cells, right? And if we are storing the glucose as glycogen, or the cells are taking in more glucose, we are therefore reducing the amount of glucose in the bloodstream. So let's look at increased blood glucose levels specifically, and this is hyperglycemia. So here we have the stimulus response model for increased blood glucose levels. So the stimulus is blood glucose levels are above our normal range. This is detected by the islets of Langerhans, which are also the modulator. So it's comparing it to our normal range and detecting that it's above the normal range. These islets of Langerhans are made up of beta cells, which secrete insulin. So this is our key hormone here, insulin. Insulin is going to act on the skeletal cells and the liver cells, which are our two effectors. So for the skeletal muscle and fat cells, we've got an increase of uptake of glucose by inserting more glucose transporters into the cell membrane. So essentially, cells are going to take in more glucose. This is going to reduce the amount of glucose in the blood, therefore counteracting this stimulus. The other effector are the liver cells. So insulin is going to act on the liver cells, and this is going to increase the amount of glucose converted into glycogen, which is then stored in the liver. Therefore, removing the amount or reducing the amount of blood glucose or of glucose in the blood, counteracting that stimulus. So this slide is just explaining what I did on the last slide in words, if that helps you a little bit more. Just remember that it is the beta cells that are producing the hormone insulin, which reduces blood glucose levels and therefore maintains homeostasis. Now let's have a look at what happens when we have decreased blood glucose or we have low blood glucose, so hypoglycemia. Here is our stimulus response model in a negative feedback loop. So our stimulus here is low blood glucose levels or levels below the normal range. This is detected by the islets of Langerhans, which are also our modulator. And so our alpha cells in the islets of Langerhans secrete glucagon, which is our hormone, our important hormone for decreased blood glucose levels. So glucagon is going to act on the effector, which is the liver cells. And the liver cells are going to produce a response, which is breaking down glycogen into glucose, which then re-enters the bloodstream. So we're increasing the amount of glucose in the bloodstream, which counteracts the low glucose levels, which is our stimulus. Again, this slide is just putting all of that into words. Just remember that it's our alpha cells producing the hormone glucagon, which breaks down glycogen into glucose, which re-enters the bloodstream and helps us maintain homeostasis by bringing our blood glucose levels back up to normal. This is just a slide or a diagram that summarizes both of these processes. So low blood sugar produces glucagon, which raises blood sugar levels. High blood sugar causes the pancreas to produce insulin and therefore lowers blood sugar. I know I just said blood sugar, but I want you to never use the word sugar when you're in a SAC or an exam. I want you to use the word glucose. A lot of textbooks or other sources will say sugar like this diagram is, but the very correct term is glucose. So we refer to blood glucose levels, not blood sugar levels. In life, most people will say low blood sugar, high blood sugar, rather than low glucose or high glucose. But in biology, glucose is the word I want you to use rather than sugar. Alrighty, so I'm going to leave you with this meme from the Amoeba Sisters again. Please write down any questions and bring them to class, and I will see you there.